Oh yeah, and another thing, I uploaded more videos onto YouTube, so we're up to date with all the live recordings on that's been uploaded. It's on YouTube. Okay, there's still some on. I mean, all the older ones are still on Canvas, but all the newer ones are on YouTube right now. Okay, so there's a playlist over there that has all the all those lectures in case you want to revisit something. Anyways, is everyone clear on that? Okay. So anyway, so yesterday we left off on talking about the liver, right? And how the liver function in a similar way to our kidneys, our renal system, in the sense that they both, that one of the functions for the kidneys is to detoxify, to remove toxins from our body, right? The same thing with the liver, right? It has different enzymes that will, that will detoxify blood, okay, in the small from the small intestines and blood, okay, the stomach and small intestines, and as well as it synthesizes bile to emulsify fats. So that means you're going to break down some fats. Now, guys, when the bile emulsifies fats, does every single bit of fat that gets does all the bile that's being used to do this to the fats, does it get lost, or do we conserve some of the bile? If you remember from last class, are we conserving bile? Yeah. So, yeah, so about 80% is going to be retained. 20% gets lost in fecal matter. Yeah, the rest of it gets stored. Yeah, that's correct, Carolina. Okay. Anyways, so we were talking about the pancreas as well. Okay, the pancreas is I told you there's two sections of the pancreas. You have the exocrine system and the endocrine system of the pancreas. Okay, all you gotta know is what enzymes exist in each. Okay, and what they do. All right, this is pretty cutthroat. It's pretty dry stuff. There's not really much more to go into here. Is everyone clear on these different enzymes? I mean, it is what it is, what these enzymes are. There's not much more to talk about. So just make sure though, for an enzyme like trypsinogen, this protein, this enzyme is the inactive form of trypsin. Okay, it's kind of like pepsinogen, right? You could think in parallel to pepsinogen. Okay, trypsinogen and pepsinogen are both the inactive forms of the actual enzyme, but they both get activated by various means. Okay, for trypsinogen, it would be enterokinase. It's another type of enzyme. It's a kinase enzyme. Okay, and then pepsinogen gets activated by hydrochloric acid's acidity. Okay, and they both actually function. Well, trypsin is going to be functioning to digest some proteins. Okay, all right, and it also activates other enzymes such as chymotrypsinogen and procarboxypeptidase. Okay, and all these three different kinds of enzymes, trypsin, chymotrypsin, carboxypeptidase, they all digest proteins. Okay, is everyone okay with this? And just as a little side note, that the way these enzymes get activated, and I showed you a picture, okay, before, they're usually changing the conformation, the spatial conformation, as well as cutting off a certain segment of the peptide so that it unshields the active site on on that enzyme, okay? It reveals that area where it's going to actually plug into the substrate, meaning its target, and the target is going to be the proteins, the various proteins it's going to digest. All right. Once you unveil that area, it's going to be able to chop up the protein to smaller bits. Okay. But before, when it's inactive, like trypsinogen or pepsinogen, they have like a certain piece of peptide covering it, or there might be some steric hindrance, meaning spatial interference, that's going to be blocking trypsinogen or pepsinogen from cutting the target protein. Okay, that's how it works. All right, and we're going to be talking about insulin and glucagon. Okay, with endocrines. I mean, we so Let's talk about it right now, actually. Okay, so let's talk about a little bit in a couple of slides. We'll talk about it. Only a few slides left, guys. It's gonna be really. This is gonna be a short lecture today. Uh, so let's talk about nutrient absorption in the small intestine. Okay. Now, at this point, whatever food that you've eaten has been digested pretty well. Okay, and in the small intestines, remember you do have some mixtures still. Remember how the food moves in the small intestine, it moves kind of back and forth through each segment of the small intestines. Well, at this point, the nutrients are going to be absorbed as monomers, meaning that they're going to be absorbing like monosaccharides, right? Amino acids, that kind of thing. Okay, that's what monomer is going to be referring to. All right, that's because it's been broken down enough at that point that's why it's so important that you want to be able that whole process between chewing up your food, okay, saliva, stomach forming chyme, okay? And then you also have when it goes back to it goes into the small intestines, you still have that little mixture churning due to the segmented nature of the small intestine. And also, the smooth muscles are involuntarily, contracting. All right, it's mixing up the whole entire whatever mishmash of food that you've eaten, right? So all that together is going to allow your small intestines to be able to reabsorb all the nutrients as monomers, which are the smallest macromolecule. Okay. And here's some lists. I mean, this is kind of a review. Okay, we have monosaccharides, amino acids, fatty acids, monoglycerides, and nucleotides. Okay, I'm gonna go over each of these again. These are all everyone should know what these are at this point. Okay. Now, hydrophilic nutrients, meaning water-loving, these are polar, okay? They love water. It's not like oil, right? They're absorbed in the small intestine and into the bloodstream, okay? Lipids are going to be absorbed into the lymphatic system and routed to the liver, where they're going to be stored, okay? And we have other adipose tissues, right? We have adipose tissue deposits around our body, though, okay? All right. That's basically it. There's not much more for me to go into this. This is pretty, dry, okay? But this I want you to look at a lot more, a little bit more, okay? Usually when I give a graph or like a nice picture, usually it's important, and I want you guys to understand how to look at it, that kind of thing. So this would be a basis for a lot of nice questions, I think. I mean, if I were a student and looking at this, I'll place some big bets that you'll get a lot of questions on this particular slide. And let's just go through it, okay? So we're going to talk about the synergistic effect on blood, okay? Talking about the concentration in glucose. Now, remember, I mentioned this before. Synergistic, you can think of it about synergy, okay? Synergy. What does synergy means? It means working together, right? It works together to increase the effect, whatever effect a pair of things have on another thing, okay? If they work together to enhance that effect, they have a synergistic relationship, okay? So we're going to talk about some of these different examples, okay? Starting with like some of these hormones, okay? Glucagon, cortisol, epinephrine, growth hormone, thyroid hormone, okay? These are all known as glucogenic hormones, meaning that they're going to be increasing the blood glucose level, okay? In other words, hypoglycemic, all right? And when you combine the effects of any of these hormones together, they're gonna have a synergistic effect, meaning they're gonna have an enhanced reaction, okay, or enhanced effect, okay? And the reason why, like I said, they work in the same way, so they have the same function, therefore they're gonna be enhancing response, and also the way it works when you have a synergistic relationships, okay, the effect the two of them have combined is going to be greater than the two individuals, um, the two individual effects, okay? Meaning that how do I explain it? When you combine the two of them together, okay, the two combined is going to have like a kind of like a multiplier of some sorts of the way it's going to enhance the effect, as opposed to having both of them kind of individually affect each other, affect the system on its own. Okay, so you have a significantly higher effect when you combine any of these together. Okay, any of the aforementioned hormone. Okay, So let's take a look what I'm talking about, the synergy, all right? And you can see how that multiplicative effect I'm talking about, okay? Where when you put one or more, two or more together, they kind of multiply in some sense. It's not simply adding two plus two. Let's say uh, glucagon gives a plus two increase on the glucose level, and let's say epinephrine gives a plus five, okay, on increasing blood glucose level. All right. These are arbitrary numbers. I'm just giving you a kind of a mathematical example, okay? If you're to just add those two together, like not add to two, if you're just to add the two together, it'll be like 14. I'm sorry, like seven, a plus four, a plus seven increase. But if you're to actually use them in a synergistic relationship, they actually multiply. So it'll be like 2 times 7 will be a 14, okay? And that's going to be expressed in the graph that you see right now. And I'll show you right now, okay? So let's look at cortisol, okay? Now, when you release cortisol into your bloodstream, okay, to increase blood glucose level, it doesn't really increase it too much. It does have an increase. It doesn't keep, it keeps it above zero, okay? It's about 70 milligrams, I want to say. This is about 70 milligrams, okay? 70 milligrams per deciliter, okay? And this is in your blood, all right, guys? Just make sure this is in your blood. Okay? Then glucagon is going to have a immediate effect, but it's not a very lasting effect, meaning that it has a very quick effect on increasing blood sugar levels, but it immediately going to decrease over time, okay? Whereas cortisol has a pretty steady and slight increase. It doesn't really change too much, but it keeps it steady above a value of zero. So it keeps your blood sugar, it's like a base of your blood sugar level, okay? It makes sure that it doesn't drop below a certain base, all right? Make sure you understand these different behaviors, okay? By looking at this chart, okay? Glucagon is going to have an immediate spike in blood glucose level, so that means there's an immediate effect on your blood glucose level, okay? Once you start to secrete glucagon. But after that immediate effect, it's going to go downward relatively quickly it goes down, okay? That means the effect of this hormone is short lasting, okay? Makes sense, guys, so far? That glucagon will be a short lasting effect, whereas cortisol kind of has a staggering effect. You get that? Trying to find the best words to kind of describe those. So let's keep moving up, okay? Now, epinephrine on its own, okay? Epinephrine, okay, has a pretty nice effect. It gets a pretty hyperglycemic effect, okay? So not only is it relatively immediate, it works pretty quickly, this hormone, as soon as it's released into your blood, but it's also going to have a slightly delayed increase, a gradual increase in of blood sugar. So that means there's a lasting effect with epinephrine, okay? And there's not much depreciation of your blood sugar level. There's a little bit depreciation after 5 hours, but not too much, okay? So it'll take some time for the effects of epinephrine to wear off, okay? Now, let's get to the good part, all right? Let's start now by combining two hormones together. So this bar here is going to show you what happens when you have glucagon and both epinephrine secreted into the blood at the same time, okay? So what's going to happen, all right? Well, like I said before, guys, you're not simply just adding it to them. It's going to be kind of like in a multiplicative fashion, okay? If you're to add, like if you're to kind of make an average of these two lines together, actually, let me just if you're to make it like add and divide these two lines together using Excel, it'll be somewhere like over here, right? It won't be it won't be any higher, right? It'll be kind of lower the line. However, in our physiology, it doesn't work that way, okay? It actually works in a multiplicative fashion. So that means you can not only, so what's gonna happen here is you're gonna be enhancing the abilities of both hormones together. So you're gonna be enhancing the ability of glucagon to have an immediate effect on blood sugar level, and also you're gonna be enhancing the prolonged and staggering effect that epinephrine has. Okay, it takes a longer time for epinephrine to get rid of its effect on the blood sugar level. So you could see that first you see a massive spike pretty immediately, right? As soon as you secrete both of these hormones into your blood. So you have a huge spike in your blood sugar level. And not only that, but you're going to have a steady increase. And this steady increase is going to be reflective of epinephrine. Okay? This is because of the enhanced ability of epinephrine by combining its functionality with glucagon. Okay? And then this spike here is an enhanced version of glucagon working by itself, okay? And this enhanced version is because of working together with epinephrine. You guys see that what I'm talking about? Is it making sense? Okay, great. Now, last but not least, and this is the best one, okay? Now, what happens when you're combining the efforts of three different hormones that have a synergistic effect, okay? I mean, the graph speaks for itself, right? You're going to be multiplying that immediate spike again, so it's even higher than before. But then the staggering effect is super enhanced. Look at that. So when you secrete all three of these together, you're going to have a massive spike and a quick increase of blood sugar level that that sticks around for a few hours, and over time it increases its effectiveness. So that means that the effects are going to be increasing. You're going to have more and more more and more glucose being present in your blood over time when you have the combined effort of all three of these hormones. Okay? Is everyone clear? So I want you to be able to look at this graph, and if I use different enzymes, but you have to understand about synergy, what synergy means, okay? If I give you a graph that shows you a non-synergistic relationship, you have to be able to identify if it's not a synergistic relationship. And the way you'll know that is by studying this one. This is a great example of how a synergistic relationship looks like, okay? A non-synergistic relationship would mean that, let's say you're secreting all three of them together, right? And if you have all three of them together and it comes, I'm going to be drawing a random line, okay? If it's going to become like this, where it's all three of them of all three of them being secreted at the same time, that means they're not having a synergistic relationship. There's most likely there's a antagonistic relationship. Antagonistic will be the opposite to synergistic, okay? There's some opposition of effects, okay? Everyone clear? And I will just write it down over here. This is antagonistic. Okay, it'll be opposite of sign. Okay, it's the opposite. That means that two or more hormones have an opposing effect on one another. Okay, make sense, guys? All right. All right, so let's talk about a little bit about glucagon secretion function. Okay. Now, these are going to be synthesized by the alpha cells in the island of Langerhans in your pancreas. So it's going to be in your endocrine area of the pancreas. Okay. So what exactly is going to stimulate the release of glucagon from your pancreas? Okay. Well, if you have a low blood sugar level that's below, and this is going to be a crucial value to remember, guys. Okay. Clinical value. If your blood sugar level falls below 70 milligrams per deciliter, then it's gonna be one of the stimuli for the release of glucagon, okay, from your alpha cells. Okay. Another one would be decreased amino acid concentration in the blood. So decreased amino acid concentration as well as glucose concentration in the blood will be a signal for the alpha cells to start secreting to start synthesizing and secreting some glucagon. Okay, so let's see what happens of the effects of insulin on body cells, muscles, and adipocytes. Okay, now adipocytes are going to be your fat cells. Okay, for the adipose tissue. Now let's see what happens. Okay, with insulin on body cells. Okay, what insulin does is one of the things is going to be breaking down glycogen in your liver to release glucose into the blood. Okay, because remember, your liver is a storage for glucose. For excess sugars, that's where it gets stored, okay, as glycogen. Okay, you form glycogen and it's stored into the liver. Okay, but once you start releasing insulin, you're gonna want to increase blood level, right? So that means you're gonna be breaking down the glycogen in the liver to make more glucose, and that glucose is gonna be released into the blood. Okay, and then what's gonna also do is gonna shift to fat catabolism. Okay, meaning that you're gonna start burning fat. In other words, you're gonna be using fat as an energy source. This is gonna be equating to burning fat. Okay, and of course, it's gonna be functioning to increase the blood glucose level in the plasma. Okay, so it's hyperglycemic. Okay, so that's gonna be insulin. Okay. I think a lot of people kind of know this already, but that's all you need to know so far. Okay, and let's talk more about our beta cells now of our pancreas. Okay, now insulin itself, guys, is going to be secreted instead by the beta cells in our Langerhans. Alpha cells is for glucagon. Okay, so alpha is glucagon, beta is insulin. All right. Now let's see here. Hold a second. Okay. All right. Sorry. So anyways, so let's, this is wrong. Yeah, that doesn't make sense. Okay, it's better. I was reading through it and I'm like, what's that's wrong. Okay. All right. Now let's talk about insulin. Okay. Now, if you know from just knowing some diabetics in general, we all know that insulin is usually an issue when it comes to someone who has diabetes or not. And so, first and foremost, it's secreted by beta cells. Okay, the half-life. This might be a multiple choice question. It's not really super important. I just have it included there. Okay. The half-life, if you remember, half-life is the amount of time it takes for half of the 50% of the population of insulin to be degraded. Okay, that's all that is. Okay, that's what half-life are. Is everyone clear what half-life is, or do you need me to mention what it is? Half-life. I mean, I just said what it was. But does everyone understand what the half-life is for? Okay, the half-life is just simply the amount of time it takes for half, 50% or half of a whatever it is. It could be a isotope, it could be some kind of radioactive material. It's the amount of time it takes for 50% of that compound to degrade. Okay? You can apply it to anything. All right? You could apply it to drugs. For example, drugs are, those are chemicals. So the half-life of those drugs, okay, is going to be the amount of time it takes for 50% of that drug to be completely dissolved, okay, or degraded. All right. So that half-life for you guys is going to be important in the near future when you start talking about pharmacology because drugs, chemicals, the drugs that have a very long half-life, that means, let's say the half-life of drug A is a day, okay? That means that person, when you're giving that person medication, that drug, they only have to take it once a day because the effect of that drug is going to be persistent because it breaks down so slowly, okay? It's going to remain in your system for a longer period of time doing its thing, okay? Make sense, Ashira? You know what? You know what? Why not? I want you guys to know that it's important. Since if a share asked me that, everyone, more than one person needs probably to know this. I mean, Carolina, she says she knows this, but if one person said no, more, it's likely a lot of others don't know this either. Okay, this is important when uh, calculating the dosage of a drug, as well as how often a person should take said drug. Okay? Let me see if I can make an arrow like this. And an increase, oh, a higher, a higher half-life equals lower dosage needed slash less pills per day. Meaning, I'm sorry, more pills. Yeah, less pills. Okay, per day. Meaning that this drug that you give this person, instead of taking it like after each meal, they'll just take it once a day. All right, maybe in the morning or in the nighttime. Okay, that's it. Okay, a lower half-life is going to be then is going to equal to more dosage slash more pills taken per day. Okay, so usually this is going to be something that uh, you know, like a lot of antibiotics, for example, those kind of drugs, those kind of pills that you take, antibiotics, and a lot of drugs you take, a lot of drugs are like this, where you gotta take them once with every meal. That's a reason why you do that is because they have a really low half-life. They're short-lived. Okay, they exit your system fairly quickly, so you have to replenish uh whatever's being lost from that chemical compound. Okay. Anyways, so let's go back. So is everyone clear on this? I'm gonna just make it red. Okay, why not? Is everyone clear on this? Make sense to everyone? Oh, ah, this I think is really important for clinical application. Okay. Uh, all right. So, anyways, so there was a mix-up, and I apologize. This is supposed to be glucagon in this previous slide. I don't know why insulin was there, and I read it, and that was my mistake. I read it off, and I shouldn't be reading it directly, but that's gonna be glucagon, okay? Because this is for this slide is for glucagon. I don't know why insulin is there, uh, but insulin's gonna have the opposite effect of glucagon, okay? Now glucagon is gonna be increasing blood sugar levels. Insulin is that you want to store away blood sugar levels, meaning that it's gonna be hypoglycemic, okay? So what's gonna be a stimulus for release of insulin from your beta cells of the pancreas, okay? Well, it's going to be high glucose levels in your blood, meaning over 100 milligrams per deciliter, or increased amino acid, all right? As well as parasympathetic activity, okay? So if you have parasympathetic activity, it's gonna be another stimulus for release, okay? Now, guys, just a question. Does insulin and glucagon, would they have a synergistic relationship or antagonistic relationship? Insulin and glucagon, is it going to be antagonistic or synergistic? Yes. Yep. So if you were to put both of them together, it would actually they would sort of cancel each other out. Like the effect. Yeah, Drew. it's going to have the opposite effect. So what's going to be doing, okay, on the body cell muscle. So the effect insulin has on our body cells, muscle, adipocytes. Remember, adipocytes are our fat tissue cells, okay? You're going to have increased glucose uptake by cells, by cell body cells, increased glycogen, protein, triglyceride synthesis. Remember, glycogen is the storage form of sugar in our body. So when we have an excess amount of glucose, we're eating a lot of sugars, it's going to be stored as glycogen. Okay. We also have more protein and triglyceride synthesis. All right. And we're going to be shifting to carbohydrate catabolism. Okay. All right. Uh, what else is there to say? Okay. Um, what else? What else I want to say? I mean, this is why when you consume a lot of sweets, a lot of sugars without working out. I mean, there's a limit, of course, but if you're someone who's just sitting around all day, like we all are right now, we're all being couch potatoes, right? And you're gonna be consuming too much Snickers or something like that. The reason why a person gains weight is because you're gonna be fostering the release of insulin, and the insulin is gonna have all these effects. You're gonna be storing more sugar as glycogen, and you're gonna be making more fat. So triglyceride synthesis mean fat synthesis, right? You're gonna be making more fat in your body, right? And as opposed to glucagon, which does the opposite effect. Glucagon is going to be increasing the breakdown of triglycerides, the breakdown of glycogen, okay? And you're going to be releasing more glucose into the blood because you need it. You're consuming so much, likely because you're either fasting or because you are working out or just consuming a lot of energy throughout the day, okay? That'll be a reason why you're going to be releasing glucagon, okay? And let's see here. So okay, this is gonna be talking about just defecation. I know this is kind of a leap. This is a admittedly poorly placed slide, and I probably would have been better to place it elsewhere. But that's it for insulin. Let's talk about defecation. Now, the actual mechanism of defecating is going to be a somatic voluntary reflex. We all know this, right? Our rectal muscles are voluntary, so we know, luckily, or thank God it's that way, right? You don't want to have a loose screw in your rear end, right? You want to be able to defecate when you reach the toilet, right? Not just like just not, you know, poop all over the floor. You know, elderly and of course babies don't have they lose well for babies at least they haven't gained full control over that. It's not strong enough, their rectal muscles, so they'll just you know poop whenever they have poop to in their colon. And as opposed to the elderly who who lose control over it because all the muscles are they're degrading, right? As you age, that's what happens. Okay. Now, it's a somatic voluntary reflex, and what's going to be involved exactly? Okay. So there's going to be impulses from our cerebral cortex. So our cerebral cortex is going to be regulating this entire contraction. Okay. So we're going to have some sensory fibers. There's going to be parasympathetic motor fibers. Okay. As well as the voluntary motor fibers. And the voluntary motor fibers is going to be innervating our rectal muscles, okay, meaning our sphincters, specifically our external anal sphincter, okay? Guys, I want to be very clear on this, okay? Out of anything to look at in this entire chart here, know that the external anal sphincter, this rectal muscle here, is the only one we voluntarily control, okay? The internal anal sphincter is involuntary, and it's also parasympathetic, okay? Now, we also have stretch receptors, okay? And also, guys, internal anal sphincter will help you defecate, okay? And how the reason why it helps you defecate is with the help of something called your stretch receptors. We have stretch receptors that kind of um, as the feces, the fecal matter travels down to your colon, okay, and it reaches your anal canal, okay. in the rectum over in this area over here, it's going to stretch out. You're going to have you have a build of fecal matter, and it's going to stretch out the fiber over here, neural fiber. Okay, all right, and this motor fiber is going to send a signal this way over here. Okay, and it's going to basically tell your anal sphincter to start contracting. Okay, the internal anal sphincter to start contracting. That's why when the ideal time for someone to defecate is when you feel pressure in near your bowels, right? You feel pressure down there. It's because of, you have fecal matter building up. And the reason why it's easier to defecate in such a time is because the internal anal sphincter is contracting also, okay? If you're only contracting your external anal sphincter trying to defecate, it's not really good. You want to poop when necessary. Or if you forcefully abuse your external sphincters, you could potentially cause inflammation of your hemorrhoids, your hemorrhoidal tissue. Right, guys? Does everyone know that? Does everyone understand that from just, I mean, maybe from your parents, just from everyday life, that if you were to stress out your external anal sphincter too much, you can cause inflammation of your hemorrhoidal tissue? Does everyone understand that? So that's why the best time to poop is when your internal anal sphincter is also contracting. And the reason why it would be contracting is because of the stretch receptors detecting the presence of a lot of fecal matter being built up in the rectum. Okay? And once it does that, your parasympathetic motor fiber is going to send the signal through the cerebral cortex and back into the internal anal sphincter. Okay? And then, of course, you have the external anal sphincter, which is just an impulse directly from the cerebral cortex. Okay. The internal anal sphincter first needs to receive information from the stretch receptors. Okay. The stretch receptors are innervating the rectum. Okay. It innervates the rectum. And once it bloats up, it's going to send a signal towards the cerebral cortex. And then the cerebral cortex then is going to relay an impulse back towards the internal anal sphincter. Okay. And this fiber, this neural fiber, is part of your parasympathetic fiber. Okay? Make sense, everyone? Now, for this slide, I want you to know what I just discussed, and I also want you to know about, you know about these two, and also know about, abuse of your anal sphincters, your external anal sphincter, like causing hemorrhoids, inflammation of hemorrhoid. Make sure you know that, okay? It's a little bit it's a clinical thing, okay? You might have some patients running into the clinic, and the nurse might be the only person who's going to be seeing you because a complication such as hemorrhoidal inflammation that could be covered by a nurse any day of the week. Okay, a doctor doesn't need to be there unless if you have a pop, if you have some kind of if it's a severe case where you need to actually surgically remove the inflammation, because something that does happen, then that whole thing could turn to a minor surgery, and that's when a doctor does it. But for normal inspection, a nurse can do it. Okay? Make sense, guys? Okay, so we will stop here. This is the last slide for today. Okay, and that's the last slide of this. The last slide of this lecture.